everyone, I'm Samantha Sharp and I'm a graduate student at UC Davis. And just getting this set up. Okay, today I'm going to present on using remote sensing to explore spatial and temporal variability of cyanobacteria blooms in Clear Lake. And my co-authors on this presentation are my colleagues at UC Davis, as well as an external collaborator from USGS. An outline of my presentation, and I'll try to be brief because I know we're running out of time, but I'll give a brief introduction to remote sensing, and then I'll talk about using remote sensing to look at the spatial variability of cyanobacteria, which answers the question of where are cyanobacteria within the lake, and then look at using remote sensing to look at temporal variability of cyanobacteria, which answers the question of when we have bloom events. And then throughout this presentation, I'll be sure to touch on the importance of this for water users and managers. To start with, to give an introduction to remote sensing, remote sensing is the process of collecting data on a subject without physically touching it. So at its most fundamental, your eyes see reflectance of different wavelengths of visible light, which allows you to collect information on a subject without touching it. And using this information, you can visually discern water from plants or phytoplankton. And this is clearly seen in these photos, which show clear blue waters versus highly productive waters. Satellite sensors measure this reflectance at specific wavelengths of light, and these fall both in the visible spectrum, which is what we can see with our eyes, as well as additional wavelengths of light that go beyond the visible. And you can see in this plot going on to the near infrared and the shortwave infrared. And the reflectances at each of these wavelengths produce a spectral curve, which you can see plotted here for different land surface types, water, vegetation, and soil. And you can use these differences between these spectral curves to distinguish between different land surface features. And you can also use these differences in reflectances to measure optical properties such as fluorescence. Early satellite missions, such as the Landsat series, measured few wavelengths of light in the visible spectrum, and we could use this information to determine chlorophyll A, which is a measure for algal biomass. But more recent satellite missions, such as Sentinel-3, measure more wavelengths of light, and we can use this additional information to calculate phycocyanin, which is a measure of cyanobacteria biomass. So we can use these newer satellite tools to tell us when we have harmful algal bloom events versus other types of algal blooms. The advantages of remote sensing over other sampling technologies are remote sensing provides global sampling at large scales to measure entire water bodies. Remote sensing allows for sampling of remote and potentially hazardous sites. And also satellite data can be used to fill historical data records for undersampled sites. Satellites provide frequent repeat samples, and remote sensing is a relatively cost-effective sampling approach. And these figures here show a picture of the Sentinel-3 satellite, and then an image it's taken over Clear Lake. And then you can apply algorithms that use those reflectance measurements at different wavelengths. And those can be used to calculate different things, in this case, calculating a measure of cyanobacteria, which is then shown across Clear Lake. So now I will talk about how we use remote sensing to look at spatial variability of cyanobacteria, where spatial variability is the patchiness of a cyanobacteria bloom. And understanding spatial variability is important for designing adequate sampling plans, informing spatial resolution requirements for future satellite missions, justifying spatial averaging of data, and all of these are important for identifying areas at risk versus those risk-free for community and wildlife uses of Clear Lake. So for this study, we calculated the spatial variability of cyanobacteria from multi platform remote sensing sampling of Clear Lake, which took place in 2019. This research has been published, and you can access this publicly available journal article from the QR code on this slide. So for this research, we deployed a drone shown in this upper right-hand photo that collected multispectral remote sensing measurements at a few wavelengths that we could use to then calculate chlorophyll A. And then we deployed an autonomous underwater vehicle, or AUV for short, which is shown in this lower right photo. Am I getting my directions right? Lower left, maybe? Sorry. And we collected optical measurements from the AUV. And this plot shows the background image is the chlorophyll measurements that we derived from the remote sensing measurements from the drone. And then the small points are chlorophyll A measurements from the AUV, and the larger points are graph samples that we collected. And the large box outlines on the plot are the 300 by 300 meter pixels from Sentinel-3. So this just gives you a perspective of what the satellite is seeing versus like how much variability you can see within each one of those satellite pixels. So we use the high resolution data that we collected from the drone and the AUV to quantify a measure called the critical scale of variability, which is the length scale necessary to resolve a bloom. So we use these plots called semi-variograms, which are basically, if you look at the distance between two samples, how different are they from one another? So samples that are close to each other are more similar, and ones that are further away are generally more different. And you can look at the shape of these plots to determine this critical scale of variability. And we use this data and determined that the critical scale of variability for a cyanobacteria bloom in Clear Lake is on the order of 70 to 175 meters. And this is important because those pixels I showed on the last plot, those are 300 meter pixels. So they're missing some of the variability within that space. And these measurements are kind of valuable to then inform future sampling efforts on Clear Lake. Another part of my research is looking at using remote sensing measurements to look at the temporal variability of cyanobacteria. And this is looking at the change in cyanobacteria blooms over time. The goals for this research are to identify environmental drivers of blooms. And of course, this is also to identify areas that are at risk versus risk-free for the community and wildlife uses of Clear Lake, which are important for end users like all of us. Why do we use remote sensing to answer these questions? It's because remote sensing tools provide frequent measurements of cyanobacteria blooms across the entire lake. So we use a remote sensing algorithm called the Cyanobacteria Index, or CI for short. And this image here is showing a screenshot from the website that you can access this tool from, which is fpad.sfei.org. The CI is calculated from reflectance measurements made by the Sentinel-3 satellite, and the data provided by this tool is critical to inform community water users and management decisions. However, in order to adequately use this, or any remote sensing tool, requires ground truthing with field measurements. So we did that ground truthing of the CI algorithm for Clear Lake using field data from our 5-year monitoring program, which Alethea presented on earlier today. We compared our field measurements of cyanobacteria biovolume, which are shown on the x-axis of these plots, versus the output from the satellite tool. And this is showing both the current CI algorithm and the original CI algorithm. And the current CI algorithm includes a secondary step, which was added in order to reduce false warnings of blooms. However, you can see in this data that it's also missing a lot of bloom events. And so these are like false positives that aren't being picked up by the satellite tool. And in fact, we find the original CI algorithm is performing better for Clear Lake. Along with CI, we use data from our water quality monitoring program to examine bloom trends in Clear Lake. We found seasonal bloom patterns with diatom blooms typically occurring in the winter, which you can see in this bottom panel of this time series plot. diatoms are shown in orange. And then we typically see that followed by a mix of green algae, cyanobacteria, and mesozoa in the summer, and cryptomonads in the fall. Cyanobacteria did not dominate in high concentrations for our time series, except during the summer of 2021 and 2022. And we found that cyanobacteria is correlated with water temperature and is inversely correlated with water level and the nutrient ratio of nitrogen to phosphorus, which we talked a lot about these environmental drivers already today, so I won't get into that. Yeah, and just to conclude, we found that cyanobacteria have high spatial and temporal variability, and understanding this is important for designing sampling plans to inform community water users and management decisions. Remote sensing is useful to look at large-scale areas such as in lakes, and also because it provides frequent sampling. But as we see, ground truthing of remote sensing tools is very important to make sure that what you're seeing is actually matching with what is actually in the lake. And I didn't really get into this because of the shortness of time, but I also wanted to mention that there are future satellite missions and also recently launched satellite missions that may provide more useful data for measuring cyanobacteria blooms. So there's going to be a lot of really exciting research to come in the coming years. Thank you. Oh.